Okay, we are at the top of the hour, so let's go ahead and get started. Welcome, everyone. My name is Cheryl Rogers, and I am the Director of Marketing at Golden Helix. Today's webcast will take a look at language disorders, and it's presented by John Eicher of the Yale Child Health Research Center. John is a doctoral candidate, and he was one of our grand prize winners in our very first annual research competition that we hosted last winter. Along with the opportunity to present his work today, John also took home a brand new MacBook Air. We will host a second annual research competition this coming winter, so if anyone out there is interested in entering, make sure that you sign up to receive updates from us. You can do that on our website. Just look for the update registration box. You'll see it on the left-hand side of the screen. And with that, I'll go ahead and pass it over to John.
Thank you, Cheryl, and thank you everyone at Golden Helix for giving me this opportunity to talk about some of the work I did for my doctoral dissertation, which I just finished here at Yale. And thank you everyone for tuning in to hear about some of my work. The title of my talk is Examining the Genetic Underpinnings of Commonly Comorbid Language Disorders, specifically dyslexia and language impairment. And what I'm going to talk about is kind of the intersection between genetics, language, using neurocognitive neurobehavioral measures, and then I'm going to touch upon some neuroimaging. But if you have any questions during this presentation, there should be a little questions pane in your GoToWebinar window. Feel free to type them in, and at the end of my talk, I'll try to get to as many as I can. If you would rather email me, please feel free to email me at john.eicher@yale.edu with any questions, and I'll try to get back to you. So today I'm going to talk about two common language disorders, dyslexia or reading disability, which I'll abbreviate with RD, and language impairment, which I'll use the abbreviation LI. Overall, dyslexia is the failure to take written symbols, in this case the word defense, split them up into their individual speech sound units or phonemes, in this case defense, and translate that into an actual concrete meaning. And my meaning, being someone from Connecticut and someone who went to the University of Connecticut, our national championship basketball team, specifically Stephanie Dolson here and Shabazz Napier, who both graduated on Sunday actually. Language impairment, so dyslexia is with written symbols, translating written language into actual meaning. Language impairment is a little different, as these children exhibit deficits in verbal language. Children cannot translate verbal utterances. In this case, my little audio file won't work on this. The word bulldog, translating that into bull, dog, and actually gaining an actual language meaning out of this. And for me, this would be Handsome Dan, our mascot here at Yale University. Both reading disability and language impairment are fairly prevalent in pediatric populations, with prevalence estimates for RD from 5 to 17%, and for language impairment from 5 to 8%. As I've mentioned already, reading disability is focused on written language, while LI is focused on verbal language, but can both be an expressive verbal language deficits or receptive verbal language deficits. I've talked about them so far as separate disorders. However, they have a lot in common. First, about half the cases of LI cases go on to develop RD later in life, and vice versa. RD cases are more likely to have had or to have LI. We say it like this because verbal language tends to develop before written language. Both have impairments in phonological processing and phonological awareness, that is, being able to separate words into their speech sound units. And conceptually and kind of broadly, these are both involve overall language deficits. Now, since this is a talk for a genetic software company, there are obviously a genetic component to my research. Twin and family studies have shown that both RD and LI have significant genetic components, with the heritability estimate for RD to be about 50 to 85%, and the heritability estimate for LI to be similar, but 45 to 73%. There have been linkage and candidate gene studies for both RD and LI, and one of the strongest candidates is the DIX2 locus and chromosome 6p22, which houses two RD risk genes, DCDC2, which stands for double cordon containing 2, and KIAA0319. Unfortunately, it doesn't have the easiest name to say for me today. I'm going to spend a little time and go into a little bit of the history of both DCDC2 and KIAA0319. Here's a schematic of the DIX2 locus and all the genes it contains. And here is DCDC2 and KIAA0319. Both have been implicated in neuronal migration, and this is shown by work by Joe Turco at the University of Connecticut, where he knocked down expression of both DCDC2 and KIAA0319 in utero during rat embryonic development, and there's impairments of neurons migrating out of the subventricular zone to the cortex. Using neuroimaging data, both variants of both DCDC2 and KIAA0319 have been associated with differences in gray and white matter. DCDC2, as I mentioned, stands for double cordon containing domain 2. The double cordon domain is a protein domain known to bind microtubules, and there's some evidence of this with DCDC2. KIAA0319 is a transmembrane protein that is cleaved, so it's hypothesized that KIAA0319 is a signaling protein in the cell. Both DCDC2 and KIAA0319 have been replicated multiple times in multiple countries with multiple languages. Now, each of these genes contains a risk variant. For DCDC2, we call it READ1, or regulatory element associated with dyslexia 1. And then there's the KIAA0319 risk haplotype in the promoter region of KIAA0319. And just to orient you, both of these are encoded on the reverse strand. So this is the 5' end of KIAA and 3' end here. READ1 is a highly polymorphic complex tandem repeat. We've actually found over 40 alleles of it. It ranges, it's about 100 base pairs long. In vitro luciferase assays have shown that different alleles of READ1 modulates activity or expression from the DCDC2 promoter. And we recently shown that READ1 specifically binds the transcription factor ETV6. Another really cool thing about READ1 is it seems to be human-specific. That is, we don't see any homology between this sequence with anything in mouse, in rat, and you only start to see it in higher order apes like gorilla and chimpanzee. The KIAA0319 risk haplotype is located in the KIA promoter into the neighboring gene TDP2. They also did, a group in Oxford showed that through in vitro luciferase assay that this variant is associated with reduced expression of KIAA0319 and this locus has been associated with both RD and LI. Now, these two genes aren't the only candidate genes for RD and LI. Another strong one is DIX1C1. DIX1C1 was found in a Finnish family back in the early 2000s, and that's actually recently been shown to be essential for ciliary motility and ciliary function. This is interesting to us because some recent papers from Europe have shown that KIAA0319 and DCDC2 may also function in ciliary function. The most famous candidate genes for RD and LI are probably FOXP2, which is found from the KE family in the Netherlands, and CatNap2. FOXP2 is actually a transcription factor that regulates the expression of CatNap2, showing a functional connection between these candidate genes. And at the time I undertook these studies a couple years ago, there had only been one GWAS examining these traits, and they examined quantitative performance on reading and language tasks in 2 population-based cohorts. So what I'm going to talk about today, I had 3 objectives. The first was to characterize the relationship of the DIX2 locus with RD, LI, and IQ. We had a discovery cohort called Avon Longitudinal Study of Parents and Children, or the ALSPAC, and went through application cohorts, Iowa LI cohort, Italy RD cohort, and the Colorado RD cohort. Second, I'm going to try to show you how I identified genes associated with comorbid dyslexia and LI through GWAS. My discovery cohort is again the ALSPAC cohort with a replication in the Pediatric Imaging Neurocognition Genetics, or PING study. And then finally, I'm going to characterize my GWAS hits using neuroimaging data found in the PING cohort. But first, we're going to look at DIX2, and we're going to do discovery in ALSPAC with replication in Iowa, LI, Italy, RD, and Colorado, RD. But first, what is the ALSPAC? The ALSPAC is a longitudinal birth cohort that took place in the Avon region of the United Kingdom. Over 10,000 pregnant women were enrolled, so there are prenatal, perinatal, and postnatal data on approximately 10,000 children. Data start during pregnancy and is actually ongoing now. The kids are actually into adulthood in their early 20s. And this was conducted at the University of Bristol. With such a long-standing cohort with so many children, they actually have a myriad of environmental and clinical data relevant to my research, including data on written language and reading, verbal language and speech, and other neurocognitive data such as IQ. So like I said, we started with about 10,000 subjects. Unfortunately, this is a long cohort, which means we lose subjects along the way. First, a subset of children did not have cognitive data at age 7, 8, and 9. So these are mainly lost to follow-up over the years. We excluded children with a total IQ of less than 75 because we wanted to look at specific language issues and not have any gross IQ deficits. Some of our samples just failed genotyping. We didn't have good DNA on them. And we also excluded those not of European ancestry because there actually weren't too many people of non-European ancestry in this cohort. And that left us with about 5,500 subjects to look at. But what did we look at with them? Like I mentioned, they had reading, language, and IQ measures in all these people, and we tried to take advantage of this wealth of data. To look at severe RD, we used the phoneme deletion task. The phoneme deletion task, children were prompted with a word such as say ball without the b sound. That would leave you with all. And this tries to measure the phonological deficits that I was talking about earlier. And children with that performed 2 standard deviations below the mean were classified as severe RD children. For moderate RD, we looked at some single word and single non-word reading tasks. There are 2 single word tasks completed at ages 7 and 9, and 1 single non-word reading at age 9. And children who performed 1 standard deviation below the mean on all 3 of these were considered moderate RD children. For language, we had 2 measures to look at. One was a verbal comprehension task. In this task, the tester read aloud a story to the child and asked and followed up with some simple verbal comprehension questions on it. And we also had the non-word repetition where children were prompted verbally with a non-word such as glack and asked to repeat the word. And again, we had 2 severities. For severe RD, children performed 2 standard deviations below the mean on either. For moderate LI, it was 1.5 standard deviations. And he also came up with a composite quantitative performance score that averaged the Z-score performance on each of these tasks together. And then finally, for IQ, we had 3 IQ measures from the WISC: total IQ, verbal IQ, and performance IQ. And we looked at these as quantitative measures. So our genotyping strategy. As opposed to just follow-up associated SNPs or associated markers from previous studies, we wanted to try to tag as much variation in the locus as possible. So we used tag SNPs, or single nucleotide polymorphisms. Basically what we did is here's the DIX2 locus. We saturated the locus with SNPs, which totaled about 200 markers covering about 1.5 megabases of the whole entire DIX2 locus. We completed our genotyping at the Yale Genomics Center of Analysis using the Sequenome platform, and we wanted to have an unbiased scan of the locus for associations with our traits of interest. For our genetic association methods, we use, not surprisingly, SNP and variation suite. This is back when version 7 was out. And we had a fairly simplistic association method looking at frequencies of the minor allele in cases and comparing that to controls. So as you can see here, the minor allele is the frequency is twice as much as in controls. For quantitative traits, we regress quantitative performance on genotype. Since we had our marker panel was filled with tag SNPs on the DIX2 locus, we built haplotypes using HaploView, using the 4-gamete rule, and completed associations with Planck. So I've only talked about LSPEC so far, but we had 3 replication cohorts. The first one was the Iowa LI cohort. This is a case control cohort of children with language impairment from the University of Iowa, our collaborator Bruce Tomlin. This is a case control cohort of 428 cases and controls, split in half. That's the number of cases. Since it was case control, same analytical methods using SVS. The Colorado and Italy RD cohorts were a little different. These were family-based cohorts of about 300 families each. Colorado is from the CLDRC at the University of Colorado Boulder, and Italy RD was from a university in Italy. At the time, we actually didn't own the PBET module SES. We do now, but for this, we used the TDT option in Plink, and we condition association on case control status. So now we get into our associations. These are our associations with RD and LI case control definitions. We have single marker here on the top path, followed by our haplotype-based associations, and a little schematic here. The order actually goes left to right, so we can follow this along and you can see the association results. First, we had a single marker associated with severe LI right here. This marker was also part of a haplotype block that was associated with severe RD and severe LI. And what I want you to notice, it's the same haplotype block, but a different haplotype. So it seems to be capturing different alleles of something or different variants of something are associated with RD and LI. We also had associations of KIA0319 and TDP2 with severe RD right here. Associations of ACOT13 and C6 or 62 with moderate LI, and one association out here in the C-Masuda gene with moderate RD. Now with our quantitative language measure, again, this was the composite measure with the verbal comprehension and non-word repetition tasks where I averaged the Z-score performance on each task. We had 2 genes showing association. First, KIAA0319, both a single marker and then 3 different haplotypes. And then 1 out here in FAM65B, a single marker out here with quantitative language. And then finally, here are our associations with our IQ measures. We only saw 1 association with total IQ, and it was way out here in what we like to call the gene desert. There aren't any coding genes out here. Then we saw association with both single markers and haplotypes with verbal IQ in KIA0319. A haplotype block stretching ACOT13 and C6 or 62 with verbal IQ, and 1 single marker of verbal IQ out here in FAM65B. So we have our discovery associations, but in this day and age, we need to replicate our associations to try to get some belief in them. And this is a very busy slide, but this is the positive replications we saw with our markers in both in Iowa, Italy, and in Colorado. And we saw evidence of replication for DCDC2 using the Iowa LI case control definition, evidence of markers in KIAA0319, and I believe in all of the cohorts, ACOT13 in Iowa and in Italy, C6 or 62 in all 3 cohorts, FAM65B in the Colorado cohort, and then some suggestive evidence in the Colorado RD cohort. So we think these associations have evidence that they are real associations. Now I'm going to go step by step and go into a little bit more detail of each one. Right, this is a schematic of the DCDC2 gene. Again, we're on the reverse strand. And what we notice is this haplotype block that I mentioned before, where 1 haplotype was associated with severe RD and another was associated with severe LI. We notice that it was close to the read1 element, which here is a little schematic here, where here is the different repeat units of read1. As you can see, they're very purine rich. And each allele differs by the number of repeat units. So some alleles have 7 copies of this GGAA, some have 8, some have 6, some have 9. So we wanted to ask whether this could be tagging variation in read1. So here are our haplotype-based association results, just to reorient you with it. So we sequence each of the carriers of these haplotypes for read1. And this involves Sanger sequencing. So there are about 225 carriers of the severe RD haplotype, and 92% of them carried allele 5, were also carriers of allele 5 of READ1. For the severe LI haplotype, which there are about 400, about 78% of them carried allele 6. And when we grouped READ1 alleles by structure and putative function, we get this clade called clade 1, which actually comprises both allele 5 and allele 6, and over 90% are carriers of the clade 1 allele. And the defining marker of a clade 1 allele is actually having 8 copies of this GGAA subunit, which is more than the ancestral allele, which has 7. And surprisingly enough, this GGAA sequence is the binding motif for the ETV6 transcription factor that binds READ1. So we think allele 5 and allele 6 introduces a new binding site for ETV6, which could change the regulatory capability of this element. So we showed linkage between READ1 and these haplotypes. Next, we wanted to actually do the association with READ1 in the full cohort. So we did someone in the lab, a graduate student named Natalie Powers, did Sanger sequencing of about 10,000 subjects in ALSPEC. And we then did associations tests by grouping different alleles for severe LI and severe RD. And what we found is there's moderate association here with allele 6 with severe LI, and pretty strong association with allele 5 with severe RD. But when we group by clade, again, with that extra copy of GGAA, we have pretty strong association with both severe LI and with severe RD. So we think this element is actually what is associated with both LI and RD. So next, I want to talk about KIAA0319 quickly. So up here, each of these black bars is a haplotype block associated with one of our traits of interest, and these little asterisks or single markers associated with one of our traits of interest as well. And we wanted to see whether these were distinct or capturing the same signal in the genome. So we looked at linkage disequilibrium, and what we found were 2 different LD blocks. One encompassing the 3' half over here, again we're on the reverse strand, and one covering not only the 5' region of the coding sequence of KIAA0319 and its promoter, but also the neighboring genes TDP2, ACOT13, and C6 or 62. So we actually think that all of these associations are tagging the same functional variants in this region, where the KIAA0319 risk haplotype is known to reside. So we had a promoter proximal variant, the KIAA0319 risk haplotype, and the READ1 element in DCDC2, which is known to bind a transcription factor. We thought that these two may interact, so we first decided to look at it genetically. So here on this graph, you can see this is the relative z-score on various neurobehavioral tasks, and up here are the different tasks. These first 5 tasks are reading-related tasks, where we have phoneme deletion, single-word reading, non-word reading, and spelling at 2 different ages. Next are 3 language tasks, the verbal comprehension, non-word repetition, and the composite task I talked about before. And here are 3 IQ tasks. So first, we looked here, these black diamonds. You can see people who carry neither risk haplotype in DCDC2 or in KIA. They perform, as you would expect, right around 0, right around average. These triangles in gray and these blue squares carry either risk haplotype, so either KIAA0319 in the blue squares, or the DCDC2 risk haplotype in the gray triangles. They perform right around average to maybe a little below average on some of the tasks. However, individuals who carry both risk elements on average perform markedly worse than any of these other groups. In fact, the magnitude is greater than the sum of the individual effects. So it looks like we have a non-additive synergistic interaction between this DCDC2 risk element and the KIAA0319 risk haplotype. Now here we're looking at the DCDC2 risk haplotype. What about specific alleles of read 1? So I'm going to show you one example of allele 5, which is the RD risk allele for read 1 and its interaction with Chi-HAP. So again, it's the same order of 5 reading, 3 language, 3 IQ tasks, and here's our average Z-score. In blue, right here, these little diamonds, individuals that carry neither are right about an average, which you would expect. Individuals with only one risk variant are a little below average, but again, not terribly below. And then, again, generally, individuals with both DC-DC2 read 1 allele 5 and the risk haplotype in KIA 0319 do markedly worse, again, greater than the sum of the individual effects. So here's a quick summary of what I've talked about so far. We have identified 4 DIX2 loci associated with read 1 LI and/or IQ. One here, the read 1 element in DC-DC2. The KIA 0319 risk haplotype, which seems to tag variation in all four of these genes right here. Although I do want to note is that we can't discount that these genes may contribute to RD, LI, or IQ. We just can't tell right now. And then we found new evidence for new associations in FAM65B and the pseudogene CMA out here. I've also shown evidence for an interaction between the read 1 risk element in DC-DC2 and the KIA 0319 risk haplotype. So far, I've characterized the relationship between a known risk locus with RD, LI, and IQ. However, I haven't really talked about anywhere new in the genome that may be associated with these. With the advent of new technology, these hypothesis 3 methods, including GWAS and whole exome sequencing, can identify novel risk genes. And now I'm going to talk about a GWAS that I've recently completed. So here, I did a discovery GWAS in the ALSTAT cohort, again, scanning the entire genome of about 500,000 SNPs, comparing allele frequencies in cases and controls. The case definitions are a little different this time, because now I specifically want to look at the comorbidity of RD and LI. So cases are now individuals with both RD and LI. And we move forward our top 10 associated markers for replication analyses in the Pediatrics Imaging, Neurocognition, and Genetics, or PING study. So who was a case and who was a control? So like I mentioned, our case definitions are a little different. We wanted to focus on persistently poor performers and on the various neurobehavioral tasks. So we had 5 reading tasks, the phoneme deletion, single word reading at 2 different ages, non-word reading, and reading comprehension. And children with Z-scores less than -1 on at least 3 of these 5 tasks were considered a case. So this left us with about 530 cases. Unfortunately, there aren't as many language tasks as there are reading tasks. However, we did have one overlap, which is the phoneme deletion task, because I thought that phonology and phonological awareness is important for both deficits, for both disorders, so I wanted to include it in both. And then I also included our good friends verbal comprehension and non-word repetition. And children who had a Z-score less than -1 on at least 2 of these 3 tasks were considered a case, and that left us about 340. So how did this break down? We had 174 children with both RD and LI, 163 with only LI, and 353 who only had RD. Our analytical strategy was pretty simple, pretty straightforward GWAS here. We set a correction of alpha equals 1.0 times 10 to the -7 to correct multiple testing. We first performed our GWAS in cases with both RD and LI, so that was about 174 cases and about 4,000 controls. We wanted to make sure that one disorder wasn't driving the associations we saw in the comorbid analysis, so we followed up by doing a GWAS in cases with only RD, so the 353, and a GWAS in children with only with LI, so the 163, in order to make sure we're seeing something specific to the comorbidity and not just something that we're seeing due to sampling error due to disorder status. And again, we performed all associations in individuals of European descent. So here are our top 10 associations for children with both RD and LI. So here's our marker information, marker name, chromosome, base pair, the minor allele, its frequency, and affected and unaffected individuals, gene, if any, it's in odds ratio and p-value. And what we see here are evidence. Well, none of our associations survive correction for multiple testing. We found pretty good association for 2 genes, the first being ZNF385D and the other being COL4A2. ZNF385D is a zinc finger protein, so it's thought to bind DNA, and has previously been associated with bipolar disorder and ADHD. COL4A2 is a collagen-associated protein, and actually has been associated mostly with cardiovascular disease and cardiovascular phenotypes. However, there are reports associating it also with white matter lesions. So we did our GWAS on these guys in the middle right here. Next, we want to go on the outside of our Venn diagrams for LI only and RD only. So first, here are our top 10 associations for children only with RD. Same kind of format for the table. And as you can probably tell right away out here, none of our associations got close to correction for multiple testing for just RD. However, what I want you to focus on is that you do not see 2 gene names here. You do not see ZNF385D, and you do not see COL4A2. So any associations we're seeing from the comorbidity phenotype doesn't seem to be driven by RD. Now for LI, again, if you look at these gene names and the location, we're seeing nothing in ZNF385D and nothing in COL4A2, meaning that those associations seem to be specific to the comorbidity phenotype. However, we do approach correction for multiple testing with markers in the NDST4 gene, and we actually see pretty good clustering of these associations. So this is pretty strong evidence that NDST4 may be associated with LI on chromosome 4. So we did our discovery GWAS. We got to try to replicate, and we tried to do that in the Pediatric Imaging Neurocognition Genetics, or PING study. The PING study is comprised of 1,300 typically developing children from 10 sites across the United States. The primary coordinating site was at a Terry Jernigan and Anders Dale's lab at the University of California, San Diego. Us here at Yale, we were one of the recruiting sites, and each of the kids underwent 3 different assessments. One was a computerized neurocognitive assessment that had many different tasks and included 2 language-related tasks, including an oral reading and receptive language task. The oral reading task, a word was presented with letters on the screen, and the child had to pronounce the word to the tester. And the receptive language task is actually called the picture vocabulary test, and anyone in the field will know it's very similar to the Peabody picture vocabulary test, where the children are presented verbally with a word and asked to pick out of 4 presented pictures which picture best matched the word. The children also went under a neuroimaging battery, structural and diffusion tensor imaging MRI, and they underwent GWAS. So here I did say there are 1,300 typically developing children. We wanted to try to match this cohort as best we could with the ALSPECs, so we only picked those who had 100% European ancestry by genetic information. So here are a replication of our top 10 cohorts covering ZNF385D, COL4A2, and NDST4 with both the oral reading test and the picture vocabulary test. We found no evidence of associations with the oral reading test. However, we did see 2 associations with ZNF385D with the picture vocabulary test that survived correction for multiple testing and are in the same direction effect, that is, they are associated with worse scores on this task. So it isn't often that you have genetic, neurocognitive, and imaging data on so many children. So we decided to follow up our associations using some of the imaging data collected, and we looked at this in 332 of the European subjects in the PING study. In all of our models, we included 5 covariates: age, gender, the scanner used, handedness, and socioeconomic status. And we decided to examine fiber tract volume and FA and 16 fiber tracts of interest. And these were all picked out of from after a literature search where we thought these may be important in language and or reading. I'm only going to talk about volume because we didn't find anything with FA actually. But before we looked at the genetics, we wanted to see whether fiber tract volumes were correlated with reading and language performance. So here on the y-axis, you have their picture vocabulary score, and on the x, you have fiber tract volume, and you actually see there's a positive correlation between fiber tract volume and vocabulary score. That is, individuals with greater fiber tract volumes did better on vocabulary scores. There's a similar relationship with the oral reading test here, although it was not as strong. But having a positive correlation led us to believe that these variables may be related, although we don't know how, and we aren't sure what it actually means. So next we looked at the association of our two ZNF385D markers with fiber tract volumes in our 16 fiber tracts of interest. And this is a very busy table, but all I want you to take from this is that all of the slopes were negative, and we seem to have widespread nominal association between our ZNF markers and with fiber tract volume. And here is just a pictorial way of seeing this with the number of copies of minor alleles on the x-axis and fiber tract volume on the y. And you see a negative slope between this. Actually, when we include global measures of not just fiber tract volumes, but global brain volumes, all these effects go away. So we think the effects actually on overall fiber tract volumes and overall brain volume, as opposed to specific effects on different regions of interest. So what I've talked about so far, we saw ZNF385D was associated with cases with comorbid RD and LI, as well as receptive vocabulary in ping. We had two unreplicated associations of NDST4 with LI and COL4A2 with comorbid RD and LI. We saw an effect of ZNF385D with overall fiber tract volume, and these fiber tract volumes were correlated with reading and language performance. So I think what both of these studies show is the importance of gene regulation in RD and LI. Rare coding variants are likely to result in more severe phenotypes, like we see in some of the other psychiatric disorders and in some of the syndromic conditions, neural conditions that we see. First, the most famous language gene is FOXP2, which is a transcription factor. We implicate a possible other transcription factor, ZNF385D, although a lot of work's needed to show that this actually A, binds DNA, and B, regulates gene expression. And another reason why we think gene regulation is important is the relationship between READ1 and DCDC2 in the KIAA0319 risk haplotype. As I mentioned before, we think that risk alleles of READ1 introduce more ETV6 binding sites, which are these little protein drawings here. So we think we introduce more binding sites here, so more ETV6 can polymerize along the DNA and change the regulatory power of READ1 and target gene expression. We are currently performing some molecular experiments to show that there's actual biological interaction between KIAA0319 and READ1, and we actually have evidence for this right now, although we need to clean it up before it's presented. And we think that this READ1 ETV6 complex could be regulating and binding to other regions of the genome. So in the future, we're currently working on meta-analyses across cohorts. As you may have noticed, our numbers for a lot of these GWASs, at least for cases, is a bit low. We have collaborators across the country who have performed GWAS of these traits in independent cohorts. So we're hoping by combining like phenotypes that we'll be able to increase our power to detect genetic associations. And then I've mentioned that ZNF385D may be a transcription factor. Before I can actually really say that, I need to prove it. So a few ways to do that is to alter expression of ZNF385D to examine its effects on gene expression genome-wide. So that could be done either by a microarray, by RNAi coupled with a microarray, or by RNA-seq technology. And to determine where ZNF385 may bind across the genome, that can be done with ChIP-seq. So since I'm giving this talk to, because of Golden Helix and to an audience associated with Golden Helix, I wanted to talk about where SVS actually made a huge difference in my doctoral dissertation work. And I think the biggest is its availabilities for and capabilities with data management. Anyone who works in genetics, especially human genetics, knows the pain of reformatting different file types or different softwares. With SBS, this is actually very easy. It's easy to import, export genetic information, phenotype data, anything you need, pedigree information, and it's easy to merge this into one file, into one data sheet. It's also very flexible in the different types of data you can use and the different options. I've used PED files, MAP files, Illumina final reports, text files, CSV files, EDI file, you can imagine, and SVS has been able to deal with it. And when it's not able to deal with the customer service and the staff at Golden Helix, have been really helpful in getting things through. I remember I had Illumina final reports for about 3,000 individuals. I was trying to get into SVS, couldn't do it. I was able to send it out to Greta, and Greta was able to take care of it very quickly and saved me a lot of months. And they're knowledgeable, lots of experience, and they're quick, helpful, and very attentive. So I just want to end with some acknowledgments. First, I would like to thank all of the subjects and families who take part in all of these studies we talked about. I'd like to thank ALSVAC, especially Sue Ring, Laura Miller, George Davey-Smith, and Beata. They actually welcomed me out there. I did my GWAS out there at the University of Bristol for 3 weeks a few summers ago. All our collaborators in our Iowa LI, Colorado RD, and Italy RD cohorts, the PING Consortium, specifically Terry and Anders, the Keck Center at Yale, who handled all the who helped us with all the sequenome genotyping of DIX2 and all our Sanger sequencing. I really need to thank Yang Kong. He developed a Perl script to call. We have to manually call all read1 genotypes. He developed a script in order to automate this. Our funding from NIH for my boss, Dr. Gruen, and myself, and the Manton Foundation. And then I'd like to thank the Gruen Lab, specifically my PI, Dr. Gruen, and Natalie Powers. Natalie designed the sequenome chip and did all the genotyping in the ALSVAC cohort. And again, I'd just like to end. If you have any questions, feel free to use the questions pane, or feel free to send me an email at john.eicher@yale.edu.
John, thank you so much for sharing your work with us today. Like John said, we're going to go ahead and answer some questions now. If you haven't had a chance, feel free to type those in the questions pane. Just a couple of quick announcements from me. The webcast will be up on our website along with the slides. You can expect them by the end of the day tomorrow at the very latest. Golden Helix is also proud to sponsor this year's Mid-Atlantic Genetic Epidemiology and Statistics Conference. That's coming up in Philadelphia on May 30th. If you're not familiar, this year's conference is focusing on the need to analyze different types of data together, which is being pushed by advances in technology as well as the lowering cost of analysis. Definitely a very informative conference for data integration, so if you're in the area, we would love to see you there. Our next webcast will be coming up on June 10th, and as we finalize the details, I will keep all of you updated. And John, let's go ahead and answer some questions.
All right, we have one question that actually asks about the functions of each of these genes. So I can talk about 3 of them, which are DCDC2, Kaya 0319, and ZNF385D. Take care of the easiest one first. ZNF385D, there have been no molecular studies done on this. Like I mentioned, since it's a zinc finger protein, we think that it may bind DNA and regulate gene expression somehow. We did look for G by G effects when we did our GWAS, and there was actually an interesting effect between ZNF385D and the promoter region of SHANK2. SHANK2 has been implicated in autism spectrum disorders, and LIN, autism spectrum disorders, at least the language component of it, are known to be somewhat similar. For DCDC2 and for KI0319, the strongest evidence are for roles in neuronal migration during early neurodevelopment. neuroprogenitor cells migrate from the subventricular zone to the cortex. When you knock down these genes, at least in rats, you don't see this neuronal migration, and they actually get stuck in the deeper cortical layers. And you actually see this for another RD risk gene, DIX1C1. There have been also, what should I say? There also have been implications of DCDC2, at least, with other neurotransmitters. I forget what specific one offhand. And there actually are some interesting studies possibly looking at spectroscopy data in humans, trying to link genes to that as well. All right, let me go through some of these other ones. What software did you use for the genome-wide association analysis? So ideally, I would have used actually SCS because it would have made my life a little easier. But since I was over in the UK did it, I actually just used Planck. But I actually did the associations as a double check with SVS and got very similar results without having to reformat all of my files. Was consenting for neuroimaging for the children difficult to obtain? For anyone who works with pediatric neuroimaging, it can be hard to get usable scans with these populations, especially for the younger individuals, because if anyone's been in an MRI, it's not the most pleasant experience. There are loud noises. You're in basically a big spaceship type thing. But we did get informed consent for all our children, and I wouldn't say it was difficult, but we sat down with both the parents and the child and got informed consent from both, and it actually helped us get good data usable data from these things, from our studies. Answer that one. And just to reiterate, someone asked if this would be available for viewing offline. I believe it's being recorded. And those are all the questions I'm seeing so far. One thing I do want to reiterate, this would be a question I had. Usually for GWAS, you need huge sample sets to see effects. We had cases in the hundreds, not in the thousands. And I think this underscores the importance of the phenotype and getting good phenotype data on all your subjects. And that can really increase the power to detect these genetic associations. If there are no more questions, I guess I can turn it back over to Cheryl. Please feel free to email me if you think of any later. Thank you all.
Thank you, John. Another big thanks for taking the time to share your work with everyone today. And thank you to everyone out there who joined us. And I hope everyone has a wonderful afternoon. And we'll see you next time.